Cell cycle control system. This is a basic overview, and this is part 2 of the cell cycle control system overview. So let us look at the learning outcomes of this session. In fact, the learning outcomes are similar to what was there in part 1. The cell cycle control system coordinates each event, giving sufficient time to each event before triggering the next event. The control system regulates, and it itself is regulated by its processes to ensure whether to halt or whether to proceed to the next event. The control system has biochemical binary switches. There are three checkpoints to regulate the cell cycle progression, one at G1 to S junction, the second at G to N M phase, while the third is at the metaphase anaphase junction. The cell cycle control is regulated by cyclin-dependent CDKs, which was the focal point in part 1 of the cell cycle control system, and APCs, that is anaphase promoting complexes or cyclosomes, which is going to be the focus in this session. And these in turn are regulated by several different proteins. So to again go through the basic principles of the cell cycle control, we know that the cell has interphase and the M phase. So the interphase has three phases, that is G1, S and G2, with G1 and G2 being the gap phases. It is during these phases that the cell prepares itself. The G1 phase, the cell prepares for the S phase where the DNA replicates, and the G2 phase is in preparation for the M phase, in during which you have mitosis and cytokinesis. So there are three basically the control system, or the controller, has a biochemical binary switch, which means that it either can switch on an event in a particular phase or it can switch off the event in a particular phase. So they work by either switching on or switching off. That is why they are called as binary phases. There are three checkpoints at transition phases. One checkpoint is over here between G1 and S, which is the start checkpoint. This is one of the most important checkpoints because the cell has to decide before going to the S phase, because once it goes to S phase, the cell is committed to cell division. Then you of course have the G to M transition phase, where the cell is going to enter the mitosis phase. And if the DNA that has been replicated during the S phase has not repaired or is not completely replicated, then it cannot enter the M phase. So here there has to be a checkpoint which is asking whether all DNA is replicated or not, or whether the environment is favorable for going into the M phase. Now, the third checkpoint, which is at the metaphase stage. So once the cell reaches at metaphase, then you have a checkpoint only if the cell is ready or has everything as wanted. So what is the as wanted thing over here? Is the question that is asked is, are all chromosomes attached to the spindle? If so, then the cell will move from metaphase to anaphase. So the third transition phase is, and the checkpoint is at the metaphase anaphase stage. So in this session, we are going to look at this metaphase to anaphase transition phase and its regulation, which is carried out by the anaphase promoting complex or cyclosome system. It is one and the same. Uh, and this APC system, in fact, once it gets active at the metaphase anaphase transition site, from there its control is still about the G1S phase. So it is able to regulate several events that happen right from the anaphase to the G1 through the G1 stage. And interestingly, the mechanism of action of the anaphase promoting complex is very different from that of the CDK system. While the CDK system relied on chemical modification, that is covalent chemical modification, that is addition of a phosphorus. So when the phosphate is added, or when the phosphorus is transferred because of its kinase activity, the substrate that is getting phosphorylated will either get active or will get inactive. So that is how the CDK system used to work, but APCC works in a different manner. So this is what we are going to look at eventually. Now we go to understanding the APCC. Uh, Please do understand that the APCC complex is quite a complex system, so we are not going to go into every subunit and its detailing, but we'll definitely take an overview on what the structure and the function of the APCC is. So, as mentioned earlier, it is called as the anaphase promoting complex or cyclosome. Now, what it has is a monomeric ring E3. This is one subunit which has a ring-like structure, and this monomeric E3 ring structure is a scaffold for another subunit which is E2. This E2 is generally ubiquitinated. Now you must understand that E2 can have more than one type, and you can have more than one E2 bound to the E3. So the ubiquitin that is present or attached to the E2 is actually transferred to the substrate, and the substrate also has a binding region in the E3, and the E3 promotes the transfer of the ubiquitin from E2 to the substrate. So the substrate will get polyubiquitinated, and this has a significance. What is the significance is that any substrate which is polyubiquitinated is kind of a mark to be degraded. That is the idea generally. So ubiquitination of a protein or a substrate is basically a kind of message that is saying that this substrate needs to be degraded. So what has been observed is that the E3 or the anaphase promoting complex functions better, or it has an enhanced function when either CDC20 or CDH1 are bound. So they act as activators. Now this CDC20 is activated by the MCDK itself. We all know that MCDK is uh, getting active when it is at the mitotic, it is at the G to M transition phase. So that transition point, that checkpoint is where the MCDK is going to get increased, and this MCDK is phosphorylating CDC20, and this phosphorylated CDC20 is activator for the E3. The other molecule CDH1, it can remain bound to the anaphase promoting complex and maintain the activity of APCC from anaphase through G1. This is something that the CDH1 has been observed to carry out. So as we've observed, the E2 is going to transfer the ubiquitins to the substrate, and the substrate becomes polyubiquitinated. Now, this is not the final complex that is there. In fact, this complex is bound to several other proteins to form a multi-subunit ring E3 APCC complex. And interestingly, it can have more than one E2 as well. And both these E2s are going to add the ubiquitin to the substrate. So eventually what the APCC does is that it ubiquitinates the substrate. There is another ubiquitin ligase complex which is called as SCF2, and this SCF is present during the G1 phase, and that also functions very similar to anaphase promoting complex. Now so we know that APCC is present when the cell has entered the M phase, but it remains inactive till the metaphase stage. Only when it is at the metaphase stage, it will become active. So the question is, how does the inactive APCC actually get active? So the MCDK, the M cyclin CDK, which becomes active at the G2M transition phase, that MCDK phosphorylates the inactive APCC and also the activating subunit, which is CDC20. So the CDC20 will have a high affinity to bind to a phosphorylated APCC. So you must understand that the APCC on getting phosphorylated is ready to get activated but is not fully active. So only when the activating subunit comes and binds to the APCC, then it becomes active. Now this APCC, once it gets active, it ubiquitinates. And interestingly, it ubiquitinates the M cyclin itself. So once you have the active APCC, the active APCC ubiquitinates the M cyclin. So you have the polyubiquitin chain formed on the M cyclin, and now this MCDK goes for degradation. So effectively, MCDK activates APC, and this APC in turn inactivates MCDK. The reason is the role of MCDK, the regulatory role of MCDK is still metaphase. So from metaphase point on, one doesn't need MCDK, and hence MCDK is degraded. Now the question is, how is it degraded? So any ubiquitinated substrate is generally guided to a complex that is called as proteasome complex, which is present in the cytosol. So the proteasome complex is made up of several different proteins, and its core is where its catalysis is. And what is the catalytic activity? The catalytic activity is that of protease. So what is a protease? A protease is an enzyme that can break down proteins. So it has two lids. This is one lid and this is the second lid. So at this lid, the ubiquitinated substrates are brought. Once it is brought over there, the ubiquitin is released and the protein per se enters the catalytic core. There, the protein gets degraded into smaller peptides and then it is released. So in fact, one should not call it as degradation, but one should call it as recycling, because any protein that goes into the catalytic core is broken down into smaller units, including amino acids, and these amino acids then make up the amino acid pool. So those then go in for production of new proteins. So therefore, it is basically recycling. But yes, the proteasome complex takes care of any ubiquitinated substrate. Now, let us look at how APCC is able to regulate. So we are not going to take all its regulatory activities, just one or two examples to show how it regulates at the metaphase anaphase stage. Now, we all know that during this S phase, the DNA gets duplicated, and therefore every chromosome you will have a homologous copy. So you call them as sister chromatids. Now what happens is that once you have the replication done, the two homologous chromosomes, or the pair of homologous chromosome, that is the sister chromatids, are actually held together. Okay, they are held together by certain proteins that are called as cohesins. Here there's a question that comes up: why do you need to hold the sister chromatids together? Because eventually, when you want to go into mitosis, you need the two sister chromatids to condense together to form the two sister chromatids, and then you have to pull them apart. Now, if you have these sister chromatids not held to each other, then they will float around separately. To bring them together at a later stage is only going to entangle every chromosome with each other, and then breaking free from each other is going to lead to breakage of the DNA, which cannot be afforded. And hence, every sister chromatid is held together by proteins that are called as cohesins. These cohesins are made up of molecules that are called as SMC molecules. SMC stands for structural maintenance of chromosome. So these molecules form a complex where you have four basic proteins. You have SCC1, SCC3, SMC3 and SMC1 coming together to form a kind of a loop-like structure, and at this point it can open and close, right? So this is going to hook up or hold together two sister chromatids. You can see that both the sister chromatids are currently in the chromatin form, but they are held together by the cohesin. Now, this is just one part. If you have the entire sister chromatid, you will find that several cohesins are bound to the sister chromatid at various points. So this is something that begins from the S phase itself and continues. Now, at the metaphase, after the metaphase, when the sister chromatids are aligned at the metaphase, you need to have the sister chromatids to be pulled to the two poles. So in anaphase, that is what is happening. The two sister chromatids that are condensed further have to be pulled towards the two poles. So at that point of time, if the cohesin is still bound, pulling the two sister chromatids to two poles is not possible. So one of the things that has to be done is that this cohesin has to be removed to have the sister chromatids together and yet not held together. So that is being aided or regulated by the APCC complex. So post metaphase, the sister chromatids, because they need to move towards the two poles, the APCC is going to activate an enzyme that is called as separase enzyme. Now, this separase is generally bound to by a protein that is called as securin. And this securin is phosphorylated by mCDK. So as long as mCDK is present, the securin is phosphorylated, but this securin, when it binds to separase, the separase remains inactive. Now, once mCDK activates the APCC and then further mCDKs are degraded, then this APCC is going to ubiquitinate securin, because the securin now is no longer going to remain phosphorylated as the mCDK is no longer active, and you can have a phosphatase removing the phosphorus from here. So the securin gets ubiquitinated, and the ubiquitinated securin then gets degraded, leaving the separase. Now the separase is active. So this separase is going to cut or degrade the cohesin. So the cohesins will be removed, and because they are going to be removed and cut by the separase, the two sister chromatids are not going to be held together, so they can be pulled towards the two poles. So you can see how a very important functionality is happening because of the APCC. Now we look at another ubiquitin ligase complex, and that is the SCF ubiquitin ligase complex. It also functions very similar to the APCC. So it is able to polyubiquitinate its substrates, and then the substrate is degraded. This is an example where we all know that CDK inhibitor proteins, CKI, so there is P27. This P27 can bind to the CDK cyclin, and on binding to the CDK cyclin, it can keep the CDK inactive. Now, when due to a certain cue, you have the SCF complex becoming active, this SCF complex is going to ubiquitinate the phosphorylated P27. So the phosphorylated P27 is then degraded in proteasome. So you can very well see that on release of P27, the P27 that was bound to an active CDK is now, the inhibition is no longer there because the inhibitor has been degraded by the active SCF complex. So many CDKs which is in the active form but inhibited by the CKI is activated due to the SCF. So it is acting as an activator for many proteins. Right. So the phosphorylation of the substrate actually over here is a cue for ubiquitination, and this ubiquitination of this and its degradation is going to lead to CDK becoming active. So to get an overview again, when the favorable extracellular environment is there, G1 CDK becomes active. That in turn leads to synthesis of G1S cyclin and S cyclin. So G1S cyclin can bind to CDK, and it can help in the cell moving from G1 to S phase. The S cyclin also, when binding to CDK, can propel the cell through the S phase, allow or enable carrying out all the functionalities that have to happen at the SCDK. We know that if the cell is not ready, then the CDKs will be rendered inactive by binding to either CKI or some other protein. Similarly, you have mCDK increasing at the G to M transition phase, and that takes care of regulation when the cell enters the M phase until the metaphase, and it is at the metaphase anaphase transition phase where the APCC takes control. And what we just looked at is how APCC can take control of at the metaphase anaphase stage. So let us conclude. One of the mechanisms by which cell cycle is regulated is by proteolysis of select molecules once their roles in the cell cycle are over. The third checkpoint at metaphase anaphase stage in the M phase is by such a proteolytic complex, and that is APCC, which regulates cell cycle from metaphase to almost G1S checkpoint. The APCC, along with activators CDC20 and CDH1, tag proteins and mediate it to get degraded by proteasomes, thereby controlling the levels of the proteins at requisite stages. The SCF is another such ubiquitin ligase complex, which along with SCF is able to mediate regulation of key control molecules of the cell cycle. So strategies of the cell cycle, especially that of the control molecules, ensure the cell cycle from going haywire. The control by ubiquitin ligase complexes has enabled the successful completion of different stages of the cell cycle. Thank you.